give new insight into embryonic research, using stem cells to build the first stages of an embryo development. Letitia Stearick. The article I chose for my topic was on gastruloids by Nancy Morris. The subject I chose was the use of embryonic stem cells to create gastruloids, which are similar to the early stages of embryos, without the ethical issues that normally present with this research. I have followed the history of embryology from the lectures to give perspectives to the growth of this field of study. Early study of embryos. As soon as scientists had the capability to study the formation of life, they did. It was not always a concise study, but nonetheless it was done in some form. Many scientists had done their own studies on different types of animals at different stages. This led to a lot of unorganized information that was not always known to those who could get the use out of it. And then it may not be as detailed as needed. Without this standardization, no one knew if what they were looking at was normal or abnormal, and at the time, there were many ideas of what may cause abnormalities, and the need to get a conclusion on what is and is not normal was needed. The need to understand what was causing abnormalities was needed also. Because of the misconceptions at the time, many felt that these had some moral reasoning behind them. So scientific understanding to put these thoughts to rest was important. This is where Johann Friedrich Meckel comes in. He started classifying normal and abnormal embryos and taking notes of the formation process of embryos. This gave others a good example of what was normal and abnormal, and a better reference to use as well. This also started a surge of new embryonic studies. But it was not quite as standardized as it needed to be for the full benefit to the scientific community. This is when Wilhelm His pushed for standardization of information to make better use of it for everyone. This brought about an increase of the study of embryology. The study of embryos. The study of human embryos has always seen controversy, and scientists in the past used the opportunities they were presented with to initiate what studies they could. With early experiments and studies, there was no ability to study an in vitro embryo or ex vitro growth of embryos, so what was used were aborted human embryos. With little data on age of embryo, the best that could be done is to categorize the stage of the embryo and if it was classified as normal. And today, we have a much clearer picture of the formation of life. Today, we also have a very clear ethical and moral guideline that limits this study to a degree. That is why scientists today not only want to make breakthroughs in embryological studies, but also in the use of new ways to recreate this process that will eliminate the ethical and moral challenges. These new developments can give scientists a real-time study of the formation of an embryo, further into the development than ever seen before. The study current embryological experiments. Today we want to study the growth of life as it is happening, to be able to understand the developmental stages as they are happening. There have been many breakthroughs already to get more insight, but if they are done with embryos, they are all limited to the 14-day rule. This rule states that research should be limited to the 14 days after creation or to the equivalent stage of development. This is a general consensus among countries and the scientific community. But this is also a limiting factor in the research that can be done. To get past this, there have been many breakthroughs to develop a way to see the progress of an embryo in real time. One of these developments is the use of embryonic stem cells to produce a gastruloid. Gastruloids are 2D or 3D models of the gastrulating embryo. This is the early stages of embryonic development. The gastruloid can be observed forming along the head-to-tail axis and turning on genes in specific patterns along this axis. This is something that has been seen in model organisms, but little about the human process was known. Since the gastruloids do not have the cell types associated with brain cells, they do not pose the ethical questions that a human embryo poses. This new model could potentially help understand and prevent birth defects and miscarriages, infertility treatments, and may be able to generate transplant tissues and treatments for other diseases in the future. Stem cells and early embryonic development. The ability to use embryonic stem cells to develop an embryo to the stages of body development. The stem cells grow to produce a changing structure that gives insight into how the body starts and develops. In vitro studies have been relied on, but this faces ethical and situational difficulties.